Hello and welcome back. Today is a bit of a doozy. We are going to cover replication, transcription, and translation, which is like all of protein synthesis. This is an abbreviated version for sure. We have an entire chapter dedicated to this, chapter 7. So this is just sort of like a precursor to what we will be learning later on. So let's get started with the central dogma. The central dogma is that you start with DNA, you go through transcription and it goes into RNA, and then you go through translation and it turns into a protein. But in the 1970s, it was discovered that retroviruses could copy DNA from an RNA sequence. So this first part right here, in which we thought DNA only could be go through transcription into RNA, is actually a reverse process as well, so you can reverse transcribe something. And so this is like why that asterisk is there, is because it's pretty recent, and so you can actually go either way. You can transcribe DNA to RNA or RNA to DNA. It's pretty commonly used now to establish gene expression, so we can determine what genes are expressed based on this particular RNA sequence. There were a number of thoughts on how DNA actually went through replication. We're going to start with replication. It has been confirmed that DNA replicates semi-conservatively, which means that when DNA replicates, it splits open, as you can see here, right? It's splitting open. And then one of the DNA strands is the old or original template, and then one is the newly synthesized DNA. So you have one old and one new semi-conservative. You partially keep the old strand of DNA. And as I said, there were like a few thought processes about how DNA actually replicated, and these were the three kind of big ones that were being tested. So there was conservative, meaning that the original parent strand would have itself, and you would just have the original strands come back together and a duplicated, brand new, all new DNA strand as a second strand after the first replication. And then as that continued, you would see that where the parent strand would continuously stay together, and you would just get duplicated copies of brand new DNA. Semi-conservative, where you have one strand that's old and one strand that's new, and then as you continue on and continue to replicate, you will get the new strand copying itself as well, which is why you see this whole yellow one, but you see the old and the new still combined. And then dispersive, in which some areas were copied from some parts of one strand and other parts of the second strand, which is why you see this almost like checkered pattern. You didn't know where exactly it was being replicated from, but you knew that it was coming from one of the two strands in some capacity, and so it was just like kind of just like a big mix throughout as DNA replicated. Eventually we were able to figure out that semi-conservative was the correct way that DNA does replicate, and we were able to figure out that using the Michelson and Stahl experiment, which experimented with radioactive isotopes of nitrogen. And so some of the isotopes of nitrogen were heavier and some were lighter, so we were able to see what was happening as the DNA was replicating, because then you could split them and you could put the DNA into a centrifuge and they would separate, because one of the isotopes is heavier than the other, so the density would show that they are resting in different areas in the centrifuge tube. And so the actual observations were that you started with a mix of the two of them, and then they split up where you still had a mix somewhat, and then you had the lighter nitrogen, which is the naturally found nitrogen in DNA. You found that as a separate band, and as the replications continued to go, this natural lighter isotope of nitrogen became larger and larger, but this mixed band never went away, which showed that you had these intertwined parent and new template strand, and as they continued to replicate, you had new strands being formed, but the original mixed strand never actually went away. And you see that the first replication, if it was conservative, you would have seen both, and we don't see that there. So kind of knocked out the conservative aspect on the first replication, and then the dispersive got knocked out later on because we saw that it did separate out to have the lighter isotope only, and that's what we see in the semi-conservative. Two big DNA replication enzymes you are going to have to know more later on, but these are the two that I'm going to ask you to know right now. We have helicase, which unwinds the helix itself and separates the strands. It breaks those weak hydrogen bonds that are between the nitrogen bases. And then DNA polymerase comes in and synthesizes the new strand, so it brings in free bases basically and aligns them to their complementary base partner, and so it allows these nucleotides to build the new complementary strand to the template strand. And then the last thing we're going to talk about with DNA replication is the polymerase chain reaction, or PCR. This is an artificial method of replicating DNA. So this is how scientists replicate DNA into massive quantities, like thousands of copies in like 4 hours. There are 3 big steps. Denaturing, so you heat the DNA to separate the strands, and then you anneal the now separated strands to allow primers that you have chosen. You choose the primers because whatever gene you're looking for, those primers will search specifically for those genes. So you cool the sample to allow those primers to connect, and then elongation, you allow the sample again to heat up a little bit so that Taq, which is just like a polymerase, like the enzyme we just talked about, you allow that to function, and it extends the nucleotide chain from those primers. So then you are again finding the specific sequence that you want to be copied, and it will continuously copy and again make like thousands of copies of whatever specific gene it is that you're looking for. So just to kind of give you like a general overview of this, this is just 4 cycles, but you do like hundreds of cycles. So you start with your template, and after the first cycle you're just replicating, but eventually you're going to find that gene that you want specifically, and it's going to be the highlighted gene that you see. And so once you see that, okay, well now I found it and the primers have found it, and we're going to start replicating that gene, and you see that it's just going to, now we went from 2 to 8, and then on this next one all of these are going to replicate into their own individual pieces, so then you're going to have even more, and by the end you're left with hundreds of copies of the gene that you're looking for, which makes experimenting and looking exactly at what this gene does significantly easier. Moving into transcription, this is the process where RNA is produced from a DNA template. The reason we need to do this is because RNA can leave the nucleus and DNA cannot leave the nucleus, and so we transcribe it into RNA so that it can be brought to a ribosome. And the way this is done is RNA polymerase separates the DNA and then synthesizes a complementary RNA strand. So the sequence that's transcribed into RNA is called a gene, and the strand that is transcribed is called the antisense. So the antisense strand is complementary to the RNA strand. Okay, so in this particular image, this bottom blue is the antisense strand, and this top blue is the sense strand. The sense strand and the RNA are identical to each other, and both of them contain the actual combination of the gene. So the sense strand has the gene, and the antisense strand has the complementary base pairs to the gene. So when the complementary base pairs find the RNA, that RNA now has the same coding as the original gene. It's very confusing. I know, we're going to practice this in class. And again, like I said, transcription occurs in the nucleus. The genetic code itself is kind of just like a base sequence of mRNA molecules. It encodes for all of the proteins that are made. They are read in triplets called codons, and each codon codes for a singular amino acid. The order that those amino acids are read in determines the protein that's produced. This is just like basic, like fast facts almost. The 4 nucleotide bases, right, A, C, T, and G, and 3 bases per codon give 64 codon possibilities. That's a lot of codon possibilities. And so that's why everybody is so unique in their own protein production, because there's so many possibilities and orders in which you can put those proteins in. The mRNA sequence always begins with AUG, and AUG is methionine, which we talked about a few lessons ago. It will always, always, always start with methionine and ends with a stop codon. You see that here. There's a stop here and another stop here. There's 3 total stops, right? 2 here, 1 here. And the stop just means stop reading, we're done, we've completed the protein. And now finally we're going to move into translation. This is the process of protein synthesis, where genetic information is encoded in mRNA and then translated into the amino acids. So this is the actual production of a protein. This happens at the ribosome, and ribosomes will bind to the mRNA and move along in a 5' to 3' direction until it reaches methionine. I was looking to see if it was in this picture, but it doesn't look like it is. So until it reaches MET, anticodons are what you see on the tRNA. So these 3 on the mRNA are called a codon. Their complementary pair is called an anticodon, just the opposite of a codon, right? And the anticodons carry the proper amino acid so that they can connect on to the forming protein. Okay, so you create a peptide bond between them. You have created a string of amino acids, which is called a polypeptide. And you continue this until a stop codon is reached. Now, a few things about the genetic code. It's universal. Almost every living organism uses the same code. And because of this, you can transfer genes between species. We have done this very successfully using bacteria. We isolate a gene. In this picture below, we have isolated the insulin gene. We insert it into the plasmid in bacteria, so we have now created a recombinant plasmid. That just means that there's 2 different species of DNA. And because bacteria can reproduce so frequently, like every 20 minutes, you can make hundreds of this transgenic bacteria. It will just continue to replicate, which means that you have copied this gene hundreds of times, and then you can extract the insulin from there. So it just makes the process of creating and producing insulin much faster and cheaper. Additionally, the genetic code is degenerate. As we know, there are 20 amino acids, but there's 64 different codon combinations. And what that means is because there's so many different codon combinations, you can have multiple codons code for a single amino acid. That allows silent mutations to occur where you can have a change in the DNA sequence, but it doesn't actually do anything to the amino acid because so many different codon combinations could potentially code for the same amino acid. Okay? Due to tRNA shape, right, you have this sort of like funky 3-leaf clover type thing. The third base is not orientated optimally for base pairing, so the third base is less discriminatory towards complementary base pairing, right? This is called the wobble position. So the first 2 will always connect, but the third doesn't always connect, and this is why the degeneracy always occurs in the third base, because it doesn't necessarily connect, and sometimes you'll get a slightly different codon combination, but luckily it's only in the third position where they differ, so you have that silent mutation, but only in the third spot. The last thing we're going to talk about are mutations, other types of mutations. So there are multiple types of point mutations where that's just one change in the DNA code. So that could be a substitution where you're swapping like one nucleotide for another, right? T becomes C. Insertion where you're adding an extra nucleotide. Deletion where you're deleting one. Or inversion where you're swapping two, right? TG becomes GT. A missense mutation occurs when the DNA change alters only a single amino acid in the chain. This is like sickle cell anemia, right, where GAG becomes GTG, and it's only one nucleotide and one amino acid change, but it totally messes up the whole protein. Nonsense mutations occur when the DNA change creates a premature stop codon, so you stop producing the protein too early. Cystic fibrosis can result from this type of mutation. And then frame shift mutation occurs when the addition or removal of a base alters the reading frame. So that means that this will change every single codon beyond the mutation, which may dramatically change the sequence itself. So this is like a huge mutation. Frame shift will affect the most amino acids out of all of these types of mutations. All right, that's everything for today. Great job, and I'll see you next time.